Okay, good morning. My name is Philip and I will talk about psychiatric-like impairments in mouse models of spinocerebellar ataxias. As you probably know, the spinocerebellar ataxias are dominantly characterized by motor dysfunctions. However, substantial part of the patients suffer also from various neuropsychiatric issues, such as cognitive impairments, depression, apathy, or anxiety. The magnitude and frequency of these issues strongly differ according to the particular type of SCA. However, in some types, the intensity, the magnitude of the deficit is very strong and may even reach the level of dementia or psychosis. It has been shown that these neuropsychiatric issues can have a very strong impact on the life of the patient, and moreover, one longitudinal study revealed that some of the issues may even accelerate the disease progression. However, we don't know what is the cause of these deficits. We can expect that the neuropathology related to the SCA may have the direct impact and may directly result in the mental issues. However, there is also another factor, and this is the emotional stress. When one is diagnosed with the incurable and progressive disease, we can expect that the person will have some mental issues, depression, apathy, or even anxiety. And dissociation of these two factors is very complicated. To get better insight into this problem, we can employ mouse models of spinocerebellar ataxias. The behavioral profiling of these mouse models is, however, surprisingly rare. This is also true for the most common SCA types, but with one exception, and this is the SCA1. The SCA1 mouse models were recently very extensively studied in terms of behavior impairments and cognition, mainly using the three relevant mouse models. The first is a knock-in mice with a prolonged alexin 1, which is the endogenously expressed, so basically expressed throughout the brain. We can call them the complex SCA1 model. The second is a transgenic mouse model where the expression of the transgene is controlled by PCP2 promoter and is just expressed specifically in cerebral Purkinje cell. We can call them the cerebral specific mouse model. And the last are complete or partial knockout for ATXN1. Cognition in the complex SCA1 model was studied from pioneer works by the Wattas et al., who firstly described an impaired learning in these mice and impaired contextual conditioning. Moreover, they found that these mice suffer from impaired plasticity in the hippocampus, what is the key brain structure in cognition and behavior. Okay, me and my colleagues wanted to go further. We therefore aim to explore the behavior deficit in the same mice in the complex SCA1 model, but in much wider perspective. So we also wanted to explore the emotional related behavior, and we also wanted to explore the relationship between these issues and motor deficit and brain pathology. We therefore compared the SCA1 mice with their healthy siblings in a variety of behavior and cognitive tests across the 4 different. We found that the SCA1 mice exhibited distinctly abnormal behavior, resembling to the mental issues in the human patients. For example, SCA1 mice showed reduced prepulse inhibition, so their startle reaction to the tone was relatively less inhibited by the weaker prepulse. In the elevated plasmase test, the SCA1 mice, at least in some cohorts, spent significantly shorter time exploring the open arms. They were mostly hidden in the closed arms. In the open field test, the SCA1 mice tend to move along the side of the arena, all together indicated increased level of anxiety. In the forced swim test, the particularly younger SCA1 mice were very passive without motivation to try to escape from the water, what suggests a desperate behavior or depressive-like. Similarly, SCA1 mice were also very inactive in the Morris water maze test, the test which is traditionally used to evaluate memory and cognition. Moreover, we also use other tests for cognition, the water T-maze test. In this test, mice are learned to find a hidden platform in the T-shaped arena. We do not calculate the latency time or distance traveled, we only record if the mice turned to the correct side or not. After 7 sessions, the position of the platform is changed to the opposite direction, so we can evaluate both the memory as well as cognitive flexibility. In this test, the SC1 mice showed relatively slower learning, but this initial learning deficit was very mild, especially in younger cohorts. However, when the position of the platforms was changed, the SC1 mice strongly struggled to relearn, so they have a very huge deficit in cognitive flexibility. Interestingly, all the psychiatric relevant deficits occurred from young age. In contrast, motor deficits and abnormal gait were rather subtle in young age and started to be very pronounced in later disease stages. Therefore, we devoted greater attention to the young age cohort when we analyzed the behavior, because the behavior results from young mice were not confounded by substantial attacks. Next, we wanted to select these behavioral parameters which the most consistently and robustly differed between SCA1 and healthy mice. So we selected these 6 behavior indicators. First is the rotor latency, the second is overall locomotion in open field. Next is the stigmotaxis in open field reflecting the anxiety-like behavior. Non-moving behavior in Morris-Walter maze test, cognition T-maze, and force swimming test immobility. We found that the rotor latency and overall locomotion were intercorrelated and also correlated with the gait parameters or the abnormal gait. So we expected that they are ataxia dependent, whereas the rest parameters did not show any correlation with a rotarot or abnormality in gait, and therefore we consider them to be ataxia independent. Next, we constructed a ROC curve, which visualized and quantified the ability of the given behavior indicator to successfully classify the mice into genotypes, so to recognize healthy versus CA1 mice. You can see that in young age, non-motor indicators distinguish the SCA1 mice with significantly higher accuracy than did the rotarot latency. So, in other words, we can conclude that early disease stages of SCA1 in mice are characterized mainly by cognitive dysfunction and abnormal behavior rather than motor deficits and ataxia. Next, we are interested in which brain regions may be implicated in the deficits we found. Therefore, we performed a volumetric analysis of different brain regions. Particularly these brain regions we suspected could have a role in the deficit and could be atrophied in the spinocerebellar ataxia. In the oldest age cohort, we identified 2 brain regions to be strongly atrophied and to sensitively reflect the genotype. The first was ratio between molecular and granular layers of the cerebellum, and the second were 2 subregions of the hippocampus. Relative volume of molecular layer of the cerebellum strongly decreased with time, so the cerebellar atrophy was extremely pronounced in late disease stage. However, in young animals there was basically no difference between the SCA1 and SCA1 mice and healthy controls. In contrast, hippocampal volume was decreased over all age cohorts, even at the youngest age cohort of 10 weeks of age. Moreover, volume of one of those hippocampal subregions directly correlated with the magnitude of some of the behavior deficits, particularly with a depressive-like behavior and cognitive flexibility. Interestingly, the hippocampus is one of the regions of the adult mammals with continuing neurogenesis, which seems to play a role in the stress resistance and cognitive flexibility. And when we mark the young neurons in SC1 mice, we found that the neurogenesis is strongly compromised. they had a fewer numbers of young neurons. But what was even more pronounced, the young neurons lacked the dendrites, which would enable the young neurons to participate in the hippocampal circuits. Finally, we look at the mitochondrial functions. The mitochondrial functions are extremely important for neurogenesis for various neuroplasticity processes and are frequently compromised in various neurodegenerative diseases. When we look at the cerebellum, we did not find any mitochondrial dysfunction. However, when we look at the hippocampus, we found that SCO1 mice has a reduced mitochondrial function. So, in sum, our results demonstrate that a complex model of SCA1 suffer from substantial behavioral abnormalities, which may be very relevant for neuropsychiatric symptoms in spinocerebellar ataxia type 1. Moreover, these abnormalities seem to be relatively independent on ataxia and cerebral degeneration, and also seem to begin earlier. Okay, recently, other team led by Maria Svitanovic and Eliza Mesher, they also studied the behavior of SCA1 mice models. However, they took advantage of using more mouse models, including the cerebral-specific SCA1 mice and ATXN1 knockout. At first, they performed another test of spatial memory using the Burns maze test, and they found that both the complex SCA1 model as well as ATXN1 knockouts showed very robust cognitive impairments. In contrast, cerebral-specific SCA1 mice showed much milder impairments. Moreover, both the complex SCA1 model as well as the ATXN1 knockouts had very profound impairment in recall 24 hours after fear conditioning. But the cerebral-specific SCA1 mice showed also some deficit, as you can see here, but it was mild. This surprisingly suggests that loss of ataxin-1 function may result in cognitive dysfunction. This is in line with results of this study, which showed that loss of ataxin-1 functions also impaired neurogenesis in hippocampus and also potentiates the Alzheimer neuropathology. Moreover, genetic studies also showed that intermediate alleles of ATXN1 and ATXN2 may be a risk factor for some types of cognitive dysfunctions and dementia. Another study of the previously mentioned team focused also on the emotional related behavior across different mouse models relevant for the CA1. Using the elevated plasmase test and the force swim test, they confirmed an increased anxiety level in the complex SCA1 model, but they didn't replicate our findings about the depressive-like behavior in the same mouse model. They surprisingly also found very similar tendency toward the increased anxiety level, also in the ATXN1 haploinsufficient mice, as you can see here. However, in cerebral-specific SCA1 mice, they found opposite tendencies. The cerebral-specific SCA1 mice tend to explore more the open arms, and they were not hidden in the closed arms. To sum up the results from studying the behavior of the SCA1 relevant mouse models, it seems that although the brain-wide loss of ataxin-1 function does not lead to cerebral degeneration or apparent ataxia, it seems to produce a cognitive and maybe also some minor behavior impairments, similarly to complex SCA1 neuropathology. In contrast, cerebral-specific SCA1 neuropathology leads to milder cognitive impairments and result to rather disinhibited behavior with a reduced indicator of anxiety. Okay, but how is it with the models of other SCA types? As I already said, the mouse models of other SCI types were rarely studied in terms of non-motor cognition or emotional-related behavior. However, it's interesting to note that some mouse models with cerebral-specific dysfunctions also showed the increased anxiety-like markers, such as this model of SCA14, And similarly, the findings from the cerebral-specific neurodegeneration model, the large mutant mice, also showed that the cerebral-specific degeneration result in increased exploration of open arms and the reduced indicators of the behavior despair. Altogether, these results collectively suggest that the neuropsychiatric issues in spinocerebellar ataxia 1 and presumably also in other types have a strong biological basis which may be partially independent of the cerebral degeneration. Cerebral deficits seems to contribute to the cognition, but the contribution of out-of-cerebral neuropathology and cognition may be even stronger. In mice, it seems that the cerebral dysfunction induced rather a hyper-exploration or behavioral disinhibition, but rather reduced anxiety behavior. So the anxiety or possible despair seems to stem from damage of other brain regions. Our results point out the hippocampus, but it may be much more complex. The studies implicate that some mental issues in the SCA1 may at least partially stem from deficient ataxin-1 function. Therefore, the future studies which aim to reduce ataxin-1 by molecular approaches, which are extremely promising as a treatment of cerebral or brainstem degeneration, will not necessarily resolve also the neuropsychiatric aspects of the spinocerebellar ataxias. Thank you for your attention.